Okay, this mini lecture is to introduce you to some of the techniques and a little bit of background for our coronavirus paper that we are reading together in class. So I thought this was kind of funny. Jojo in your class alerted me to some of these coronavirus memes. So anyways, let's talk about the real virus. So you have a wiki on SARS and coronavirus that talks about the virus, the pathogenicity of it, its replication, its genome. So please look through that as some of your background information. What this paper is focusing on is basically how the virus mutated to be able to infect humans. And this paper is specifically looking at SARS, coronavirus, which was a really big deal in 2002, 2003, and 2003, 2004. A little bit more, but not quite the epidemic that we had in 2002. As you know, we have a new coronavirus out that started in 2019, and the same kind of analysis is being done to try to understand how this virus jumped from animals to humans. So, schematic of the virus, and what we're really talking about in this paper is this S protein, the spike. And the S protein shown here has two components, an S2 and an S1. And so the majority of the paper focuses on the S1 subunit because that is what is going to interact with the receptor. And the receptor is called ACE2, and it's a functional receptor in human and other animal cells. And functional receptor means it has another function. So the virus takes advantage of this receptor, but the receptor does have other functions in humans, has been studied. And this is just a reference. What I thought was interesting is it shows tissue expression of the ACE2 receptor. And what's really interesting to me is hardly anything in the lungs. So coronaviruses cause respiratory tract infections. Here is just a screenshot of some of the papers that have been done to characterize the functional receptor, so what it does normally in humans. It's an enzymatic receptor, which means it can cleave proteins, and they think it can regulate cardiovascular and renal functions. So you can see, looking at some of these titles of these papers, the paper we are reading did cite Hemming et al., and so I went to look at this paper because since it said the cells that were expressing, what was the quote from the paper? Hold on, I underlined it. Tissue expression of the receptor corresponds to localization of virus in infected individuals. And we've talked about tissue tropism, right? The viruses only infect certain types of cells, cells that express the receptor. And so when I first looked here, I thought, well, why is it affecting the lungs? But then researchers have looked more microscopically, and this is lung tissue, and they're showing staining for ACE2. So they said it's clearly present in alveolar epithelial cells, so those cells in the little sacs of your lungs, capillary endothelium, fibrotic lung tissue. So ACE2 is expressed in the lungs, which allows the coronavirus to infect the lungs and cause respiratory illness. The other thing I wanted to mention is within the S1 protein, somewhere there is an RBD, which stands for receptor binding domain. So my suggestion with this paper and with all papers that you read is you kind of keep maybe one sheet of definitions or explanations for all these different acronyms and cell types and proteins and spike proteins and things like that, so you can do a quick reference. Sometimes it's hard to keep track of everything. But they will be looking at this specifically at the receptor binding domain, which actually binds the receptor more strongly or with higher affinity than the whole full S1 trimeric protein. And I call it a trimeric because you can see there's 1, 2, 3 protein subunits that make up that S1 subunit. Okay, so we know this is a zoonotic disease. Coronaviruses in general are found in bats. So SARS was actually identified in horseshoe bats, but then they seem to jump or move to different animals. So the intermediate, we think, between bats and humans was this cute little guy called a palm civet. And I feel bad that he's in a little cage here. So here's one in the wild. And here they also mention a raccoon dog. Oops. That's this little fluffy guy right here. And so what we think is the big issue is palm civet to humans. And one of the reasons this little guy might be shown in a cage is palm civets are actually kept in markets. So there's different live animals in these live animal markets that you can go and choose the animal that you want to eat for dinner that night. So there were, they believe, in the market where the SARS coronavirus came from, they think it came from live palm civets in this fresh meat market, and that probably in the butchering for meat of these palm civets, a person was infected. So that's zoonosis, right? Going from animals to humans. There are 3 different coronavirus spike proteins that this paper is going to look at. Excuse me. So TOR2 is the 2002-2003 SARS coronavirus with a 10% mortality rate, which is really high, and that's why it was such a scary infection. The 2003-2004 SARS coronavirus was much less severe, and the spike protein, the S1, is called G2. And then the palm civet from the animal, the S protein, is called SZ3. And in this paper, they're looking at one, how this SZ3 S1 changed to become this TOR2 and perhaps become, we don't know which way it went, the G2 S protein. And so they're looking at changes for the S protein because that's what interacts with the receptor. And they're also looking at differences between human cell receptor and palm civet receptor because both, all animals use this ACE2 receptor, or I should say all coronaviruses use the ACE2 receptor to infect. And they noted 2 to 4 changes between the human, I don't want to draw it like that, 2 to 4 changes or differences between the human ACE2 and the palm civet ACE2 allowed this SARS coronavirus to infect humans. So there's differences. I don't want to say changes because these receptors are not changing themselves. They've just evolved with different amino acids. But the real change comes from the virus gaining a mutation that gives it an advantage that it can infect a different cell type, different host. Rat cells, the ACE2 receptor from rat cells was used kind of as a control. Rat cells are not susceptible to these different coronaviruses, not very efficiently. And so in the study, you'll see that they make mutations in the rat cell receptor to mimic human cells or to mimic palm civet to map out exactly which are the essential amino acids to allow the spike protein and the receptor to interact. So let's talk about some of the techniques. So one of the techniques that's used a lot is the idea of chimeric proteins. Okay, so chimera is a mixture. And so what they're doing is they're taking the gene, say from the human and the civet, right, which makes their own proteins, and they're mixing pieces of them together. And these can be single amino acids. These can be a short 4 to maybe 10 amino acid changes. But you get what we call a chimeric protein, which is a mix, or maybe not a mix is the good word, but some human, some civet or rat, depending on what they're doing, amino acids. So this is the way that they are mapping the important parts of these different proteins. And they also do it with the S1, right? They'll change or mutate some of the amino acids, or I should say the coding sequences for the amino acids, to make an S1 chimera. Okay. So that's all done at the molecular level. So you manipulate the DNA, which you know then expresses the protein. So change or mutate DNA, you get a new protein. Just trying to see where I'm going. Okay. Another type of chimeric protein that they're making is they're taking the S1, so this is the spike, S1 subunit, and they're putting it on to the bottom part of an antibody. And this is strictly so that they can do a technique called immunoprecipitation, which I'll talk about here. So here's a normal antibody. The two Fab parts are specific for antigen binding, and so those are different for every antibody. But the FC, the constant region, is the same. And so we can buy commercial antibodies that recognize this, and so we can see what S1 is interacting with. So I'll show you immunoprecipitation in just a sec. They also put tags on the ACE2 receptor, right? So receptor is a transmembrane protein. They put different tags and they have these different antibodies, 1D4 or 9E110, that recognize the tag. And the tag is just a series of amino acids a lot of times around 9 or so, that are added to a protein to allow for analysis. So this is totally artificial. It's just a way that we can have an antibody bind to this tag so we can see what the receptor interacts with. So the big picture here is we're looking at protein-protein interactions. So one way to do that is called an immunoprecipitation. Whenever you see the word immuno, this means antibody. Precipitation means to pull out of solution. Okay. So they're going to use an antibody that's specific. Antibodies are very specific to look at protein-protein interaction. Okay. So for instance, the antigens here. One could be the S1 and one could be the receptor. And we use an antibody to see if these two are interacting. So you could use an antibody to the receptor, or you could use an antibody to S1. Then you have something called protein A cephalose, which is basically a bead with antibodies. So we always draw antibodies as Ys. So you can see that here. Antibodies look like a Y. And so this has antibodies that bind the antibodies. So these are specific for the Fc regions. And so they can pull down antibodies that maybe have proteins interacting together. So this is how we do the precipitation. And then the washing step means you just wash out all the other stuff that didn't bind. Elution means you release all these proteins, and then you run something called a SDSPAGE or a Western blot. Okay. So the whole point of immunoprecipitation is that you have a specific antibody, so you can look at just specific proteins. And here we're looking for protein-protein interaction. So if I had assigned this better, let's see if I just change this a little bit. If I said this was S1 and this blue was the receptor, then I could say, let's see if S1 interacts with the receptor, right? And we would have these in our sample that we are analyzing by SDSPAGE. The more interaction you have, the more protein you're going to, oops, what's going on? The more protein you're going to pull down, and we'll talk about what that looks like in just a second. Okay, so SDSPAGE is used to separate proteins based on size. So in these experiments, they had either cells that they were breaking apart and getting all the proteins from, or some were artificial in vitro in a test tube, mixtures of proteins to look at interactions. So basically, after they did the immunoprecipitation, we call it an IP, they loaded these different samples. So each well is a different sample. And then you apply an electrical current and the proteins move based on size. So smaller proteins move faster, larger proteins move slower. And if you looked at all the proteins, there would be thousands, and you just see all these what we call bands. So the band represents molecules of the same size. So sometimes what they'll do is do what we call a Western blot, where they'll use antibodies to detect specific proteins within all of these bands. What they did in this paper was they radio-labeled or metabolically labeled the cells. And so what that means is you put in radioactive, so you saw, if you look, you have 35S, what else did they, and a methionine, I can't remember which, was it 35 methionine also? Just give me a sec. Yeah, of course I can never find anything like in the moment. Oh yeah, 35 cysteine, 35 S-methionine. Okay, so, sorry, cysteine and methionine. So 2 amino acids. So you make the cells radioactive, and so all the proteins that the cells produce are radioactive, assuming they have a methionine or a cysteine amino acid in them. And then once you do the immunoprecipitation and you run your different samples, the dark spots that you see, the bands, are actually radioactive. So you use film to detect it. You don't need to understand the details of it. This is kind of an older technique, not used as much. We've tried to get away from using radioactive materials in research, but it's very sensitive, so it's good if you're looking at maybe low expression. So what's happening again in the cells, let's say, let's look at here. So what's happening in the cells is they have these cells called HEC293 T cells, and they'll put in a plasmid that, say, makes a recombinant receptor. So this is my chimeric receptor. So you take the plasmid, this is the DNA that has our chimeric gene sequence. And the process of getting the plasmid into a cell is called transfection. So they transfect the HEC293 T cells with this plasmid, and it will make this chimeric receptor. Okay. Now, if they put radioactive material in here, then the receptor is going to be radioactive. And then what they're doing is they will take, say, the S1 protein that they've modified, look for interaction, and use an immunoprecipitation where they're using an antibody to S1. They pull that interaction out, they run it on a gel, and the only thing you're going to see is you're going to see a band from the radioactive receptor in this case. So they do a couple different techniques, but most of this is immunoprecipitation. And so they can make all these different chimeric receptors, and they wouldn't have them all obviously in the same cell, but this would be 3 different experiments. So I just want you to understand that you change the DNA in a plasmid, you transfect it into these human cells. These are what they look like in tissue culture. You can radio label them, and then you can run an SDSPAGE and look and see if there was interaction. If there's no interaction, right, then when you ran that sample, it would be blank. In case you're curious, HEC293 T cells are a cell line, which means they're used in lots of labs, grown kind of forever. They have some cancer characteristics that make them immortal. They're from human embryonic kidney cells, and the T stands for a large T antigen. And the reason these cells are used, not that they have anything to do with a coronavirus infection, but they're easy to grow, they're easy to transfect, and the T for the T antigen allows for a high level of protein expression. So this is just a totally artificial system, but it's allowing us to look at these protein-protein interactions. Another method they talk about in this paper is called flow cytometry, or FACS. And so the idea here is you have a population of cells or molecules. So it's heterogenic, meaning you have multiple different types. And you can put it through flow cytometry, where there's a laser that will read each individual cell, and it can basically count. So it can quantitate the number of cells. And so one way you can do this is with an antibody, and you can see how many cells does that antibody stick to. You can do it with, they talk about GFP, green fluorescent protein. So green fluorescent protein will shine green when it's excited by the laser, and you can count how many cells are expressing GFP. So I don't know exactly why they do flow cytometry sometimes instead of the immunoprecipitation. I'm guessing that maybe the flow cytometry is a little more sensitive. The other bonus of flow cytometry is it's not radioactive. And as I look through this paper, I'm wondering if they had multiple, probably graduate students or postdocs working on this project using different techniques, because as we'll talk about, they use different pseudotyped viruses, and here we talk about they use different techniques to detect these protein interactions. I don't totally know why, but that's my guess, is that multiple people were collaborating on this, each using kind of their best method. So, as I mentioned, they have pseudotyped viruses. So this is if you want to ask, does infection happen? So before, we're just looking at protein-protein interactions, but what we really want to know is, can this changed spike protein allow for a virus to infect? And so we have pseudotyped viruses, which are also like a chimeric virus. And they have both MLV, GFP, and HIV luciferase. So these are both retroviruses. And that what they can do with this virus is put an artificial coronavirus spike protein. Okay, so the idea is we change this spike protein to coronavirus, and then we see if this virus can infect cells that are expressing the ACE2 receptor, and maybe one of the chimeric ACE2 receptors. And then to tell if infection is happening, you can measure the amount of green fluorescent protein. Right? So if a cell is infected by this little virus, green fluorescent protein will be expressed. Or if the cell is infected by this recombinant or the pseudovirus HIV with the S1 spike protein, right? You measure luciferase, and luciferase is measured by a chemical reaction. It's very sensitive to like low levels of expression, or GFP is used by the FACS flow cytometry. So the whole time, whenever you see that they're using pseudotyped viruses, the point of this is to see if infection happens. They also talk about surface plasmon resonance, and honestly, I don't know how this works. All I know is that this is more sensitive. It helps enhance looking at these protein-protein interactions. And they use it to determine the dissociation constant, KD, and this is in the table. And I really don't understand very much about enzyme kinetics, but what I do know is that a higher KD value, so a bigger number, means there is lower affinity for those two proteins to stay together. Okay, so the smaller the KD value, so it's like 10 to the minus 7, is going to mean these proteins don't dissociate very well. They stick together really good. And if you have, say, a 10 to the negative 4th, that means they're falling apart a lot easier. So keep that in mind when you're looking at table 1. But like I said, I don't really understand how this works. Okay, so this paper is all about proteins, protein structuring, and numbering. So I just want to give you a little bit of a reminder about protein structure. So in red here is shown alpha helices. So this is just part of secondary structure of protein. These ribbons represent beta-pleated sheets, another type of secondary structure. And then you might have these connecting. So the whole, all these lines are a chain of amino acids that make up the proteins. And so like this would be a loop connecting an alpha helix and beta-pleated sheet. All these numbers refer to the amino acid. So we start at the N terminus of a protein and we just number the amino acids 1, 2, 3, 4, 5, 6, 7, 8, all the way to the C terminus. Okay. So this is showing you, this is amino acids 12, 13, 14, 15. So they're very near the N terminus. These are amino acids 101 to 107, right? So if you stretched this out, you would have 1 till, I don't know how many there are in this one, let's say 200, right? And that's how they're numbered. And the numbering is important, especially for us, because they talk about changing specific amino acids. So you might want to keep this chart around. This is telling you the amino acid and its one letter symbol, so they kind of go back and forth between these. So for instance, you might see K479N, which means the lysine at amino acid position number 479, so it's the 479th amino acid, was changed to, if I could spell was, excuse me, to N, which is asparagine. No, I shouldn't do it like that. ASN. Sorry. Aspartic acid is ASP, asparagine is ASN. Right? So this is how you interpret what all these letters mean. The original was lysine, and now it's asparagine. So they're going to change amino acids, or they call them residues, between the human and the civet sequences. The last thing they talk about in figure 7 is they talk about glycosylation. All I want you to remember about glycosylation, it means you have sugars added to the protein. And hopefully in cell biology you learned that this happens in the ER and the Golgi, depending on the glycosylation. This is just giving you examples of different types of sugars or molecules that are added on to proteins. They don't talk about what type of glycosylation it is, but if you have a protein, let's say our protein here, and on one of the amino acids you stick a bunch of sugars on it, you can see that if another protein was trying to bind, it could block it, right? Glycosylation can interfere with protein interactions. So it's important to know if the region of the protein you're looking at is glycosylated or not. Okay, so that should help you with techniques from the paper. Even though this is a 2005 paper, I want you to know that they're still looking at this. So this paper just came out on the 19th of February, and they just figured out the structure for the new 2019 coronavirus spike protein, and they're looking at the interaction of that with the ACE2 receptor. So it maybe will help us someday develop a vaccine if we can figure out a very conserved part of the spike protein that blocks interaction with the ACE2 receptor. All of this data doesn't really help us right now with treatment or prevention, but again, maybe looking at conserved regions, we could maybe design a vaccine against coronavirus one day. So, like I said, even though our paper is a little bit older, they are still doing the exact same work with the new coronavirus that just came out. Okay, I hope that helps you work through. It's going to take you a lot of time to work through this paper and these figures. Give yourself time, give yourself breaks, and then we will be discussing it in your small groups. All right, thanks.